It's a pleasure to introduce Rudolf Jaenish as the keynote speaker at this conference. Rudolf is a founding member of the Whitehead Institute and a professor of biology at MIT in Cambridge. And as most of you probably know, Rudolf has been interested for a long time in understanding the epigenetic basis of development of cell fate transitions in different contexts and its dysregulation in diseases. And he's been using very different systems over the years, ranging from exon activation, neural development, pluripotency, and more recently cellular reprogramming by nuclear transfer and transcription factors to dissect these questions, and has really made seminal contributions in each of these areas. And has obviously also received a lot of awards and rewards for these contributions, which are way too many to remember them all, but one of them to mention is the presidential medal, which he received last year, I think, received by President Obama. So one of the things that I find very impressive about Rudolf's research, from being a student in his lab and following his research before and afterwards, is his ability to identify important biological questions that have been very difficult to address with conventional approaches, and then develop new tools and technologies to rigorously and in the best possible way resolve these fundamental questions. And I think this goes back in his career in terms of developing the very first transgenic mice and the first knockout mice ever to study the role of DNA methylation development, and spans all the way to more recent years when Rudolf adopted and developed designer nucleases such as zinc fingers and talents and CRISPR technology to probe gene function in cultured cells and in mice in vivo. And I think we'll hear a little more about some of these topics in his talk. So thank you very much for coming, and we look forward to your talk.
So thank you, Konrad. And it's really fun to be here in Cold Spring Harbor. It was a great meeting. So what I would like to talk about is about, so what we heard yesterday also from Lawrence, really disease modeling. And I really want to talk about two topics. One is gene targeting, and the second one is autism. So you might ask, what do they have to do with each other? Very little is the answer. So there were two different issues. And I will really keep my introduction rather short, because I don't think you need introduction to the field. So obviously we know the system of iPS cells and ES cells, what is very useful. And I just want to point out that if you want to make disease modeling a reality by differentiating patient-specific cells to the effector cells, or to use cell therapy, one of the things we have to resolve is efficient gene targeting approaches. So that's what motivates me to summarize some recent approaches. So a challenge in disease modeling, of course, to find an in vitro phenotype. And over the last few years, and this is a very incomplete list, quite a number of phenotypes have been published, which are ascribed to patient-specific conditions. As for conditions like schizophrenia or others, which I think is amazing that one finds a cellular phenotype. We're interested in Parkinson's, and for a number of sporadic Parkinson lines, we have derived there was no phenotype whatsoever, which may be not unexpected, because it would be expected to be very subtle. One of the key issues is, a key challenge is, what is the right control you're using? And we heard before about the differences and the difference in differentiation potential between iPS cells and between ES cells. So when you find a phenotype in your patient-derived cell and not in the control cell, you have to wonder whether it's due to system-immanent iPS variation or due to the cellular phenotype. So a number of years ago, we made a decision really to overcome this. We want to make isogenic pairs of cells that differ exclusively at the disease mutation. They come from the same embryo in case of ES cells, or from the same fibroblast in case of iPS cells. And so the idea would be to either correct a mutation in patient-derived iPS cells, or to introduce a mutation into ES cells, with the goal to to derive isogenic pairs of cells which only differ at that nucleotide which is disease-causing. So let me just briefly summarize what we did for the Parkinson with Synuclein. Synuclein, there are a number of point mutations which give you a dominant, give you early onset of Parkinson. And the goal was, we really were interested in these two mutations, A53T and E46K. And the goal was to make isogenic pairs of cells differ exclusively at one of these two nucleotides. So either by introducing these mutations into ES cells or correcting this in an iPS cell. And the strategy was either using a vector which carried the mutation to insert, or, which I will only talk about, is to using a single-stranded oligo which carries the mutation. So that's what we did. And just summarizing this, these are the two mutations. And the cells were transfected with a single-stranded oligo containing either this or that mutation. The cleavage, zinc finger cleavage, was at the AT53 mutation here. And then the question was, with the same cut, could you also insert the base pair change 30 base pairs upstream? And you can. It's about with a 1% efficiency here. So you go through a lot of work to do that. But once you have these, you have cells, you don't leave any footprint. No LOXP site, nothing's left. So they're really, if you want, pristine cells which only differ at this one mutation. So if you get the phenotype in your experimental cell, you have some sort of assurance it may be due to the disease you want to study instead of the variation in the system. And so this, of course, is fine if you use monogenetic diseases. You can, if you know the gene, you can make those. But of course, it gets much more complicated if you look at polygenic diseases, where there are many genes change, giving a small effect. I think that is a problem. These are, of course, the more important diseases. So this approach really has a limitation. And the major limitation is it's really valid for monogenic diseases, but really the most important, as I said, are the polygenic or sporadic diseases. So a potential solution, which really drives some of the efforts, would be if you do it more efficient, gene targeting, gene exchange, whatever manipulation, could you solve that? And that's what brings me to the CRISPR-Cas system. This is the clustered regulatory interspace short palindromic repeats. So let me introduce the system briefly. It is an immune system from bacteria, where bacteria have a locus, a CRISPR array, which has small 20 base pair repeats here, which correspond to sequences in invading phages or plasmids. So upon infection, they activate this locus. The RNAs get processed to hairpins. The hairpins hybridize to the invading plasmid. They're complex with a protein, and this protein then cleaves and sort of kills it's an amazing system. So if this is the viral DNA, it's called the protospacer here. These 20 base pairs is flanked by the PAM sequence, the protospace-associated motif. And this is the host DNA, which doesn't have the PAM sequence. That gets activated. You process the RNA. It complexes with Cas9 in the type 2 system. And Cas9, this complex, then cleaves the invading DNA. And so the question was, could you use that like the talons or the zinc fingers to target genes maybe in multiplexed way? So this was experiments done by these three. Shikdu Shivalila is a graduate student, Hayu Wang, and Hui Yang, a really very efficient team. So we targeted TET1, TET2, TET3, because we had done this all by conventional targeting, and that's what took us 3 years to do that. And so we knew the system. So the idea was then to transfect TET1, TET2, or TET3 together with Cas9 DNA and pick clones with selection, and then ask the question what the targeting sequence frequency is. And so the strategy was, if there was correct targeting from the guide RNA, then you would destroy a SAC1 in TET1, an ECOR5 in TET2, and the ZO1 site, endogenous site in the TET3 gene. So another strategy is to really destroy these endogenous sites, which I don't think is the best argument. So everything was confirmed by Southern and by sequencing. So in all of the experiments I tell you, I do believe in Southern's a lot, were confirmed. So what did we find? So when you do that, and this just shows you these clones, you find actually a very high frequency of homozygous mutations. The frequency, 70 to 80% of the S-cell clones have homozygous mutations in the targeted gene. So this is pretty high when you think about what you achieve with conventional ways. So this then prompted us, can you do it simultaneously? Can you really target 3 genes at the same time? So we use TET1, TET2, TET3. And I show you now here just the RFLP analysis for triple knockout clones. They're all, this is the wild type band. They're all destroyed, the relevant site. And this is confirmed by Southern. You can see the wild type band has disappeared. And they are, of course, they're different deletions, so you get different bands up here for all of these. And you can sequence the 2 alleles, and you know that you have target both alleles because they have different deletions. So, for example, this is an insertion here and deletion here, and this allele, this is 2 different types of deletions, and so on and so forth. So these are triple mutations and triple homozygous mutations with 1 transfection. So what's the efficiency? The effect is what you expect. If you knock out all the TETs, this is our conventionally double knockout, TET1, TET2. Took us 2 years to make that. These are these triple knockouts, and you can see some do have some activity in this not terribly accurate assay, and some have none. I think one thing is clear. You don't know whether you have a null or a hypomorph because the deletions from non-homologous end joining are unpredictable. So they have all reduced 5 HMC levels, exactly what you predict from the conventionally targeted null mutations. So what is the efficiency? The efficiency is, if you just do the triple knockout, is about 20%. So basically, 20% of simultaneously transfected cell clones carry homozygous mutation in all 3 targeted genes. So with one shot, you get 6 mutant alleles. So simultaneous targeting of 3 genes is quite efficient. Why not using 5 genes? So we did 5. And we used, in addition to TET1, TET2, TET3, we used 2 Y-linked genes, UTY and SRY. Those genes on the Y have never been targeted by homologous recombination. The Y seems to be quite resistant to that. And so when you do that, and here I give you the results, efficiency, clones with 6 target lanes with more than 50%, 7 was 50%, and actually UTY was sort of less efficient than all the others. 10% carried all those. So 50% of the ES cell clones simultaneously targeting 4 genes generate cells carrying mutations in 7 alleles and 10% in all 8 alleles. So you can do it with 5 genes. I think there's no reason why you can't do it for the dozen genes. So we don't know if we haven't tried more. So this is pretty efficient. So do you want to avoid, I mean, ES cells is still a lot of work. You have to do chimeras, germline transmission, takes a lot of time. So can you do directly in the fertilized egg? The approach was then to co-inject Cas9 RNA and guide RNAs directly into the zygote, develop them to blastocyst, and generate mice. So this is the idea. You inject those, TET1 or TET2, together with Cas9. And what you get is, I show you here the mutants. This is, again, the Irflip analysis. They were all mutated in both alleles, and the Southern confirms that. And of course, here the sequencing also confirmed that both alleles were targeted. So can you do it simultaneously, again, like in the S cells? So this would be double mutant. It was actually even more efficient than before in the single ones. Actually, there was 1 heterozygous animal here. So this is a wild type. 1 was heterozygous. The rest were all homozygously targeted. And here you see, I think there was no non-targeted. These are postnatal mice. This was all confirmed by Southern. You can see the heterozygous one indeed has 1 wild type band, and the other ones are all targeted. And you get mice. So what is the efficiency for this? So we use 2 different concentrations, TET1 and TET2. There's not much toxicity, as you can see from the cleavage to blastocyst. There are clearly a lot of deaths. And that we know, because the double knockout of TET1 and TET2 really gives you a certain fraction of the pups die during embryogenesis, so that's we expected. So the key thing is of those which survive, really basically up to 80% of these animals at the targeted zygote stage carry homozygous mutations in both genes. So with double knockouts in 3 weeks. So we took 2 years to make the double knockouts with the conventional targeting in ES cells. So that's quite a safe, efficient way of manipulating mice. So, of course, what you do this way with the cleavages, you don't know what you get. You get a deletion or insertion of unknown size. So can you make predetermined mutations where you really know what you want? So I talked about the point mutations in the Synuclein gene using talons or zinc fingers. So can we do it with CRISPR? And so the experiment here was then to also inject 2 oligos, which carried a point mutation in the 2 genes, right, close to the cleavage site. And the approach then was co-injection of these RNAs together with an oligo, and then the idea was, if this was correctly targeted, you destroy the endogenous site and you create a new one. So we can really see clearly correct targeting. So if there was correct targeting of this oligo, you would destroy the endogenous SAC2 site and create an ECOR1 site, and for TET2, you destroy the ECOR5 site and create an ECOR1 site. So this was the criteria for correct targeting, and it really worked well. 60% of the mice, of the postnatal mice, carried the point mutation in both alleles, in both genes, in one allele of both. I show you this one example here. So this would be the example here of these double oligo-targeted mice. The wild type here, of course, is cleaved by SAC1, and you can see most of them, more than 80%, have really lost the SAC1 site, and those which have lost it, some of those have now gained an ECOR1 site, and this is, of course, a wild type which doesn't have an ECOR1 site. And you see the same thing for TET2, where all of them have lost the endogenous site, and some of those which have lost it have gained an ECOR1 site. And if you sequence those, then you can see exactly that in these double engineered mice. They have ECOR1 site in one allele, the other one has a mutation. This apparently possibly is homozygous for the ECOR1 site, or it has a very large deletion, which we haven't determined. But we couldn't see anything else, and these were also heterozygous and had a deletion on the other allele. So there are 3 ways to make mutant mice. You can target ES cells, up to 5 genes, probably more. You can get very efficiently direct targeting in mice, in the zygote, and you can induce predetermined mutations also in the zygote. So this work was published in May. And so let me summarize. So in ES cells, up to 5 genes, 50% efficiency, 1-step generation of mice with homozygous mutation with 80% efficiency in all, in both genes, 2 genes, predetermined mutations, 60% efficiency. So there were quite a number of unresolved issues. That's what I want to come to. One is it useful for marker insertion by homologous recombination? Can you make conditional mice in 1 step? Can you make defined large deletions? What is the off-target rate, cleavage rate? And I give you the update. The update would be we inserted reporters into 3 genes, SOX2, NANOCO, and OCT4. We generated conditionally mutant mice with defined deletions. We did a careful off-target analysis, and we looked at the mosaicism. So those are the issues I want to go through. So first, SOX2, we target with a very short tag, V5. It's good antibodies. You can target it very nicely. So it was inserted. This is the guide RNA. This is stop codon here at the end of the SOX2 gene. So this is the guide RNA here. And you can see here that the pups in general had insertion in 1 allele, not in the other one. And this one was a homozygous one, this one. And only that one, if you use this one here for Western, this has a signal with the antibody, but not the wild type. And it stains correctly in the inner cell mass or in ES cells, which we derive from targeted embryos. So about 30% of these embryos or ES cells carried the correctly inserted tag. The NANOC, we targeted with a larger piece, 700 base pairs, again at the 3' end. It was fused into the reading frame with a 2A sequence, and the southern cells was correctly targeted, and the expression was, again, as you would expect, in the inner cell mass no, this is no GFP. It was in ES cell clones, but when you differentiated the clones, the ES cells, the signal disappeared, as you would expect from a correctly targeted NANOC gene. So the efficiency for this one was by 7 to 10%. And finally, the biggest insert we did was 3 kb, just off the shelf. We had one of these constructs used previously. It was 3 kb long, so it's really it has the GFP and has then still the selection cassette, which we didn't remove, so it was 3 kb. And again, the southern short was correct targeting, and the blastocysts were stained really were GFP positive in the inner cell mass, and the ES cells derived from them were also positive. So in this case, the efficiency was 18%. So if I summarize that, the efficiency of those, and here you see the number of embryos injected was quite a lot for those, more than 1,000. The toxicity is really not really a concern, the way we did this. But this is the key issue here. These are the correctly targeted mice. You get between 8 and 30% of the manipulated embryos carry the reporters insert an endogenous gene. So basically, 1 day injection, and you will have in 3 weeks, you will have your mice. So that's, I think, is certainly really a useful speeding up of these experiments. So with conditional mutations. Now, in conditional mutations, you have to get 2 inserts on the same allele, right? So we used MECP2, which I will talk later much more about. And the idea was to delete exon 3. So we had made several guide RNAs on each side, and we choose of the 2 of each side, L2 on one side and R1 on the other. They're cleaved better than L1 and R2. And so correct targeting was, of course, all confirmed with Southern. Now, MECP2 is an X-linked gene. So male should have 1 allele, either mutant or wild type. And the female should have 2 alleles, either mutant or wild type, right? Now, when you look at this here, this is a, which male? This is a male here. The last one has 2 alleles. That's a bit funny. And this one is a female, which has 3 alleles, right? And I'm going to go to the other map. So it turns out, so these 2, of course, they're mosaics. So in this case, it's clear that the insertion of the LOXP sites occurred after the zygote stage. So you have a mix of both, which is common in traditional transgenic pronuclear injections. Now, are these alleles functional? If they're on the same, of the 2 LOXP sites on the same allele, and this female number 1 had really 2 alleles, two alleles, and you can see upon CRE, you delete 1 out, and there's the circle which you get. And the male, of course, had only 1 allele. That is male number 3. This guy here had only 1 allele, and that you delete out. So it's functional if it's on the same. So what's the efficiency? So these are the number we injected, and you can see about 50% of the mice bone have an insertion. You can also see that females are more efficient than males, of course, because they have 2, and the target size is double. You find that L2 was more efficiently inserted than R1. Well, there was just efficiency. We don't know exactly why. That is not totally clear. We had animals which have the LOXP sites on 2 different alleles. Now, for males, of course, that was not expected, so those are 2 mosaics. But more importantly, many more than on 2 alleles had it on 1 allele, right? And these were the functional ones. So you can expect, at least for this gene, 16% of the mice had the LOXP site inserted on the same allele. They were conditional and they were functional. So that was certainly a satisfying result. So can you make find large deletions, because I said these deletions you get with the cleavage are really unpredictable. And so for this, we use two guide RNAs, which are spaced by whatever you want to space them, and no donor DNA. So we used again MECP2. We wanted to delete cleanly exon 3. And so we did this, and exactly what you get, those are cleanly deleted, 3 of those. And you can sequence this. Here's exon 3, and these 3 guys have deleted all exon 3. And of course, the ends are not defined because of non-homologous end joining. So, but you clearly, they're nulls. There's no exon 3 left. So these were mice with 7 base pair defined deletion. So we're wondering, where do you inject this? And all this, what I told you, was cytoplasmic injection, which is not the way traditional transgenics were made. They're pronuclear injection. So this was a cytoplasmic injection. We used different concentrations of DNA. Too high concentrations are toxic, so we reduced that. But then we thought, and these are the numbers I showed you, and then we thought, well, if you inject both into the nucleus, and of course we reduce the DNA and everything much more, and you get about the same efficiency. And then we thought, well, we should be fancy and inject in a 2-step, the RNA into the cytoplasm where it belongs, and the DNA 2 hours later in the nucleus where it belongs. So we did that, and that was really a big failure. So nothing happened. So for us, the optimal conditions would be those. The efficiency is the same, right? So not you have to reduce, use 200 nanograms of DNA, and this was sort of optimal for us. There's no difference really in the efficiency, except pronuclear injection needs some skills. You have to find the nucleus. Cytoplasmic injection is really trivial. So that makes it really very simple. So off-target. Now, the off-target mutations was a big concern. There were 2 papers published in July in Nature Biotech. And this one, for example, is concluded here that even up to 5 mismatches, so they have a certain target sequence, and they looked at all loci which have now mismatches, 1 to 5 in this case, they have a target higher than the intended target. Now, this was really of big concern, right? And another paper really came to a very similar conclusion. So that was a big concern, so we decided to really look carefully at the off-target cleavage. Because that's what I mean, was seeing this probably unuseful, not very useful, this method. So the criteria for off-target analysis was we used 7 different loci, 7 different guide RNAs in 6 different genes. We looked at all potential targets up to 3 base pair mismatches. We used the surveyor assay, which is exactly what used also in these papers, right? It's a mismatch assay. And any mismatch we got, we sequenced to be sure we didn't hit a polymorphism. So the off-target cleavage, to give you an example, SOX2. So this is the PAM sequence, and the guide RNA is really numbered 1 to 20. 20 is the most 5' of the guide RNA. And these were the 13 off targets with 3 base pair deletions. And none of those was cleaved. So that was reassuring. Now, NOC, you had 2 off targets. One had a mismatch at position 20, and more than 70% were cleaved. So this is not a very discriminatory mismatch. The second one had 2 mismatches here at 19 and 18, and one in the middle. None of those was cleaved. So let me just summarize all these data. So we tested, as I said, 1 to 3 mismatches in 7 different loci in 39 mice. We found 3 off targets. One was the one I showed you in NANOC, 1 mismatch at position 20. One was an OCT4. It had 1 mismatch at position 19. This is not very discriminative. And the third one was MESP2, and one of those, 10% cleavage, 1 mismatch at position 21 and 10. So clearly what comes from this is mismatches at the 5' are not very good to discriminate. So I think from this we will conclude there's a high specificity of sufficiently high to use a CRISPR system. I'm not sure if it's as high as talents. The analysis has been not done as carefully, but I think it's absolutely high enough for anything. And I'm happy to talk later about how we interpret the difference with these published data, with our data. So mosaicism is high. 40% of the animals we get are mosaics, which is probably lower than traditional pronuclear injection. They in general most of them tend to insert after even later. So I talked about cleavage and mutations. Can you use it to activate genes without changing the genome? So can you use it as the guide RNA to target a sequence you're interested in, a gene you're interested in? So for this, you would use a Cas9 mutant, which is catalytically inactive, wouldn't cleave, but still guides, still recognized guide RNA. So this is the idea, of course, what I talked about. You have the RNA, which guides, and the Cas9, which then together this complex cleaves. And so if you use now a mutant Cas9, which cannot cleave, and you bind a repressor to this, it has been shown you can silence a target gene. We were interested more in the activation of a silent gene by fusing an activator to this. And this work was done by Albert Cheng, who was a graduate student in the lab. So what he did is the following. He made a cell line which has a transgene TD tomato, which is driven by a TET operator. And this can be now activated two ways, either with a reverse transactivator, which also has fused 3 VP16s. And this can be activated by giving DOX, so positive control. And then the other one is the Cas9, also fused to these 3 VP48. And this will be activated by the guide RNA. That was the idea. So this is transient transfection, this whole thing. And when he looks at this, and you probably cannot see that, the red here, but it's pretty, the DOX, positive control, you give DOX to get this pathway activated. You find 80% of the cells become fluorescence. This is the negative control. This is no DOX. And when you don't use DOX, but use now the guide RNA, it is 60%. And actually, this was not totally controlled because it was a transient transfection. There could be the transfection efficiency could be different. So I think it's very efficient in both of those. So that was good. And so the question, can you use it for an endogenous gene? So this was in 293 cells. We used this gene, ILR1, which is not expressed. And he made a number of guide RNAs upstream of the promoter. And he used them all together. And the activation was not really useful. There was very little. So he changed strategies and made another kind. So if 3 VP6 are not enough, why not 10? So he made it what he calls Cas9 VP160. And then he targeted this again to the ILR1 locus. You see here the guide RNAs in the upstream of the promoter. And what you can see is that guide RNAs 1 to 3 are really sufficient to 6-fold activate the gene. And if you all of them, it's a little bit more. So really the proximal ones really give you a reasonable activation. And for SOX2, we did the same thing. And again, you find 10-fold activation. But it's interesting. Guide RNAs which are in the gene really inhibit. So if you use 1 to 5, these 2 and these 3 here, really you don't get activation. But if you use, for example, 3 to 5, those, you get good activation. And the best activation, 3 to 7. Right? So that gives you a 10-fold activation or so. So clearly binding of guide RNAs close to the promoter gives optimal activation. When you downstream, that seems to be inhibiting. Upstream, close to seems to activate, and further upstream seems to have less effect. So what are the applications? So I think generation of genetically altered animals in one step, carrying multiple mutations, define predetermined mutations, conditional mutations, reporter mice. And I think the technology will be useful in species where you don't have ES cells, where this will be the only way to do it. Right? I think it will be really transform those. I think it's really, for people who work in the mouse, it is a game changer. I think to make mutant animals, it makes ES cells basically obsolete. I think it's just a detour. In human, ES, iPS cells, any type of mutations or gene correction, I think we should be able to make. You can make isogenic cells in monogenic, maybe in polygenic diseases. You can put reporters on key genes. I think we need that to develop differentiation protocols which are efficient. I think it's useful for activating endogenous genes for many applications. And I think, since targeting is so efficient, I believe that's will be the way to really target and gene correct somatic stem cells, like let's say hematopoietic stem cells, so which are very rare. And we know only something like lentiviruses can get into hematopoietic stem cells, whereas some other issues cannot. So if you have the right gene delivery, and there are good viral gene deliveries which are not integrating, which you probably don't want to integrate, I think that will be an interesting application. Okay, that's all I wanted to say about gene targeting. Let me now switch totally to autism. So autism. There are quite a number of sporadic and genetically defined autisms, and I'm going to talk about RETT. RETT is due to the mutation of MECP2, which is a global transcriptional repressor. It is 100% penetrance, and it's a very high incidence, 2%. It's probably the highest incidence of all autism. So that's what we're interested in. Autism, the onset is at 1 year of age. It's only in females. It's an X-linked gene, as I said. Boys die in general. Very severe mental retardation, no speech, stereotypic hand movements. They sit and wring their hands. And the anatomical sites, they have a smaller head growth, and they have smaller neurons. That will be important later, as me pointed out. And so we made a mutation more than 10 years ago, also Adrian Bird, and the results were very comparable. The mutant brains are smaller, as you would expect. They have all sorts of defects in nocturnal activity. And when you look at sections in the brain, the mutant neurons are smaller, significantly smaller than the wild-type mutants. And this is really an amazing model. It really mimics incredibly well the human condition. For example, the mice even wring their hands, their paws, like as I showed you with normal mice spread it. These guys don't do this. So it's quite amazing. So what's clear from quite a lot of data is this is not a developmental disease. It's a disease which is postnatal. And this comes from, I think, really a very nice paper from Adrian Bird, and we did it a little bit differently, where you can re-express the endogenous gene in an adult mouse which has symptoms, or we did it from a knock-in, and you reverse the phenotype. So clearly, you need MECP2 expression all the time, but it's reversible. These are very severe symptoms you have. And then there were two other attempts to rescue those, either by totally different pathways, by BDNF expression, it was transgenic, that relieved the symptoms, or by a small peptide, IGF1 peptide, which can go in the brain, and that also can reverse RETT-like symptoms to a certain extent, and this is actually now in clinical trials because it was a FDA-approved drug. So the idea is MECP2 is a repressor. That's what everybody believes. It binds to methylated CpGs and recruits repressive complexes. That was a dogma. That was the idea. The problem was, when you look in the mutant mice, this didn't fit. So when we looked at, and that was no more than 10 years ago, when many people confirmed that, looked at microarray data, there was just not really a global upregulator. It was really flimsy. It was really not really consistent with the hypothesis. So we want to do this now in human cells, and it was driven by Yoon Lee, and Hao Wang again made the mutations with talents. And so they made with MECP2, they deleted exon 3. I talked about exon 3 all the time to generate isogenic pairs of mutant and control cells, which also had a GFP, which is useful. And so we wanted to see what's happening in these human cells in culture. And so clearly what you first see is if you compare male and female cells we have, then, and you differentiate those two neurons of 2 months or so, they're clearly smaller, significantly smaller, as we've seen in the mice. When you look at neurites, and you can sort of quantify this on a grid, then the mutant, female and males, so the W3 is a female, this is a male, so we use it all the time. They have significantly fewer neurites, and they're shorter. You can see here for males and females. So they really have a cellular phenotype, these cells. And maybe interesting also, they have a very interesting electrical phenotype. So you can plate these cells in a microchamber, which is a microelectrode chamber. We can measure electric activity. So it's one chamber. Up here are mutant neural precursors plated. Here are the controls. And you have to differentiate those for 2 months. That's a problem. So you have to keep them not contaminated. And you can record. And you can see there's a lot of activity, spontaneous electric activity in the wild type, but very little in the mutant. So it's a very drastic difference. So that's visiting at the expression, this expression thing. And when we did this, we did the usual conventional microarray. It was just what we saw in the mice. There was no difference. It was a little bit of upregulation. So this was, again, in male and this in female cells, and they were differentiated for different times. So this is totally consistent with previous results, with MECP acting as a weak, really weak, maybe global repressor in neurons, but really not what you predicted. So let me divert briefly. Gene expression and cell size matter. And this comes from a paper from the Young lab, where they looked at MYC target genes. Now, MYC has been profiled in hundreds and thousands of studies because it's important for clinical applications. You want to know what MYC does. Now, what Rick realized is when MYC is overexpressed in cell, the size increases. So what they showed here is when you do the conventional way, where you normalize your Affymetrix for the input RNA, you get a certain activation or silencing of genes, but you compare really 2 different situations. A cell with a lot of RNA with a cell with little RNA. And depending what your expression difference is, particularly if it's medium or lower, when you now compare this cell by cell, so you have the input of the cells is the same, then you find potentially very different results, right? And that's what they published last year. So that was, of course, very interesting. And so we looked at the MECP neurons, the mutant neurons. They have much less RNA. As you can see, the neural precursors have the same, but upon differentiation, they really lose a lot of RNA. So the experiment was then to take neural precursors, differentiate those, and at different times collect exactly the number of cells from controls and from mutants, extract the RNA, you add a spike in to correct for any loss after that, and then you do your microarray. And what you find, using now the Young lab approach, was really quite different. It was striking. You might see it. So in males, less prominent than in females, you find a global, actually, inactivation of genes and very little activation. So when you do this on a show here, then you can see that there's no difference in the neural precursors, but in the 4-week neurons, there's now a significant inhibition of gene expression, less so in males. So according to this, MECP acts as a global activator in neurons, but not in neural precursors. So that was just the opposite was what the concept was. Of course, that is very consistent with a paper which was published last year from Ned Heinz's laboratory, where they showed, very surprisingly, that MECP binds to hydroxymethyl. Now, hydroxymethyl in the gene body, of course, correlates with gene expression. So that, again, didn't make sense with MECP being a repressor. So that makes perfect sense now with being a global activator. So this is consistent with its role in gene activation. So what are the target genes? So when you look at this, then you find that glia genes are not very much affected, as you can see here in these different situations. But synaptic genes are. They're the ones which are affected by the lack of MECP2. And immediate early genes, FOS and so on, they are really highly affected. They are really the ones, the target genes, which are affected by the lack of MECP2. And when you just look at the low expressed genes, the middle, higher, and the top expressed genes, they're looking at the level of expression, then it's clear that the top genes, the highest expressed, they are most affected. Whereas the ones which are very low expressed, they're really not affected. So it's really targeting, apparently, affecting the highly active genes. So what actually is most affected, important, are ribosomal proteins. They are really affected. They're much more affected than the overall genes. Ribosomal proteins, and when you look at protein synthesis, that is not affected in neural precursors, but it's affected so significantly in neurons. I mean, I showed you the RNA, because the RNA was mostly ribosomal RNA, so this is really consistent with protein being down. And when we look at mitochondrial function, I don't show you the data, mitochondrial function is impaired. So, of course, the mTOR pathway is key for cell growth, or size of growth, and it's activated by all these input expressions. And what I told you before is BDNF and IGF1 are known to increase protein synthesis via the AKT-TOR pathway. And what I told you before, those were shown to rescue part of the RETT phenotype, because they would bind here, right? BDNF IGF1, this little peptide. So we asked the question, we exposed these cells after 2 weeks differentiation to either BDNF or IGF1, and then looked for soma size. We can see is both of those tend to increase the soma size, not quite to the normal size, but substantially. And this is for both cell lines, for the male and the female line. So you activate the AKT pathway, and you rescue, at least in part, the cell size difference in the mutants. Well, there's another way to activate this pathway by knocking out P10, which then would increase AKT and S6. So that's what we also did, is to confirm that. So we used the lentivirus, which expresses P10 in shRNA, and cherry to mark the cells which were infected. You can see here there are quite a number of cells which are infected. And when you now take, for example, those out here, then you can see that you stain for cherry, then these two cells were infected. This cell was not infected. This cell has P10. These are the not. So we have an efficient knockdown of P10. That's what's the only thing. So what happens when you do that? And to summarize that, first of all, you really nicely increase the cell size when you do that. It's interesting. The neurite arborization, this would be the mutants, and this would be the ones which with the knockdown really goes up to really increases in complexity substantially. And protein synthesis is really restored to more or less almost the wild type levels. This would be the control if you use rapamycin. If it goes down, of course, as a negative control. So P10 inhibition, BDNF addition, or IGF1 treatment rescues defects in cell size, in neurite complexity, and in protein synthesis. So that's the idea. In wild-type neurons, wild-type MESP2 would bind to genes which are methylated. So it binds, clearly shown, to methyl MC, and probably silences those genes. But it also binds to active genes. Namely, it binds to 5-HMC of active genes as an important for expression. In the mutant situation, where you don't have MESP2, then you have a limited upregulation. We see that. There's some genes, some set of genes is upregulated, but really mostly have a global downregulation of active genes. So we come to my final slide then. So in normal neurons, you have MESP2 here. You have a certain level of RNA and protein. And in the mutant cells, you have smaller cells. They have less RNA, less protein. But from all what I told you, there's a way to restore, make a phenotypic rescue by inhibiting P10, basically by activating AKT, and probably by BDNF and IGF1, which possibly goes also over the same pathway. So I think it's really gives us a new, at least for me, a new way to look at these at Rett syndrome and these rather complex changes you see in the brains, and it's amazing that you can restore function by adding these simple, affecting these simple pathways. Okay, thank you very much. when we come to the last slide, I mentioned most people, Hai Wang was really driving the CRISPR-Cas system and also the talent, Hui Yang and Shiktu, Shivalila, also Albert Cheng, I mentioned him, Milad Davlati was in the TET experiments, Frank Salt and Dirk Hockermeyer was involved in the original zinc finger experiments, Yun Li and Julian Muffat were the drivers of the RETT experiments. We collaborated with Rick Young on this, and with Miguel Casu at the Picor Institute also on the RETT experiments, and really the initial zinc finger experiments and telling experiments were done together with Sangamo. Thank you.
Thank you very much, Rudolf.
What are your thoughts about the recent studies that showed that the defect in RETT may be corrected by through the neuroimmunological cells, such as microglia, so replacing the microglia, and so that refocuses.
Yeah, I think that's clearly. So I think it's maybe not a cell autonomous disease only, but could be. So I think we can test that. We can make sort of glia cells from these mutant or other cells. We have to mix and match, and we haven't done that. So I think that's clearly shown in the mouse, and I think it's very interesting.
But also in many respects, doesn't it kind of make us a bit more cautious about the types of cells we focus on, because we focus on one cell type as if it's an end to it all, and whereas there are other influences.
So in this case, we really look at neurons. So we have really not looked at the glia influence. I think that needs to be done. Yeah.
Rudy, the fact that if you normalize to gene expression, you don't see any changes, but you normalize
to RNA input.
To RNA input, but when you normalize to cell, you see changes, suggests that there's a proportionate to cell size mechanism for global transcriptional regulation.
Right.
So have you looked at other times when cell size changes and see if there's global proportionate changes? Like, it would be interesting if there's some fundamental mechanism.
So in the same experiment, in the null precursors, you don't see cell change, and there was no difference seen. So we haven't looked carefully at this, but I think it's very important because certainly in the mixed studies, I think people made the wrong conclusions, and this was pretty important. A lot of implication of those. So I think they have to be redone now. So from that point of view, I think Rick's observation is significant.
So Rudy, you gave this beautiful mechanism by which you, with various different tools, you could recorrect the phenotype. And I'm wondering, given the pathways that are affected, which are more general pathways, are there ways in which you can then take this to your advantage to try to treat these patients?
Well, I mean, you have to, for the patients, you have to get them into the brain. So IGF1 is this very small peptide, it's 5 amino acids. That goes to the brain. So that is ongoing, and I don't know the result. In mice, it worked. So you got a amelioration of the phenotype, which was significant. So I think that has to be seen if that works in patients. BDNF, that is tough, right? You can't get BDNF into the brain, that just doesn't work. Can you do it with other, I mean, inhibiting P10 might be a little dangerous, right? So there might, that's an issue which I don't know. So can you turn on MECP2 in the brain? Can you activate the MECP on the inactive X, right, by something? I think these are all issues which people are interested in, and I think that's certainly an interesting possibility.
Are there specific ways to target the neurons in question or target the cells in question? Are there mechanisms for, I don't know the field at all, but what kinds of mechanisms might be used to target target? It's not clear
what cells are responsible for the phenotype. Yeah. It's totally, all cells are affected, all neurons are affected. So in our cultures, mostly the excitatory neurons, but I mean, we look in the brain of mice, all neurons seem to be affected which express MECP2. So it's known that patients get in these crises, autonomic crises. So they get in hypoventilation, suddenly the heart stops. So really, these are emergencies, you have to go to the hospital, otherwise they die. So clearly, these centers are affected by this. But I mean, this doesn't explain the behavioral phenotypes, because all neurons seem to be really affected in a major way in neurites. And so it's a puzzle that you can restore that. I find this amazing. You can restore the function even after you have this network which is so defective, right?
Do you think there's any selectivity in terms of which proteins are depleted, or is it really just a global ribosomal?
So we don't know whether they're specific ones, but you saw it's global, and it's really the mitochondrial proteins are particularly affected, and the ribosomal proteins, which of course consists with a decreased protein synthesis. Whether a specific one, which do know the early response genes are particularly affected, or the highly expressed genes are affected. So all this points to this is pretty global. What's happening? These neurons are lacking something very important. It's interesting that BDNF can restore part of this. Now, BDNF, of course, affects survival, certainly. Survival is not an issue in the rat. There's no cell death. So I think it's a puzzle at this point.
Yep. Have you done whole genomic, as for the CRISPR, have you done whole genome sequence mixture like off-target, especially when you put some...
Yeah, we have not done this. Nobody, as far as I know, done this. It's very expensive, right? But it probably should be done.
Here? Yeah. Very excellent work. And I want to know, in your last slides, the rescue experiment, did you do any functional experiment like electrophysiology? Because you showed that there is change in the morphology.
Right, it's a nice experiment. We haven't done that. Yeah, it would be nice to also see that, but we really haven't done that.
Yeah, question regarding the first part of your talk, and obviously your lab have tried all the the FN technology, tooling, and CRISPR. So can you comment that? Do you think that the first two technology is dead in the research field?
No, I think so. I would think zinc fingers, I would not use them. I think they are, in a way, probably they're too complicated to make. So they seem, to my opinion, other people have a different opinion, they're obsolete. I think talents and CRISPRs, I think, are complementing in certain ways. For example, CRISPR is so efficient. When you make a insertion, I think most of the time you will have the other allele mutated. In talents, in our experience, that is not so. So you insert something like in the Synuclein we did, only in 30% the other allele is mutated. So it could be that those are quite, they could be useful for different purposes, both of those, I think.
So how about off-target? So do you think CRISPR is better than?
No, I think the CRISPR off-target analysis is not as extensive yet as it was done for talents and zinc fingers. But I believe, so we have these two coming back to the difference in the published data, which was so concerning, right? Why is that? Why do we not find it and they find this enormous high off-target rate? So I think there are a couple of reasons. They used human cancer lines, which are repair deficient. So that is a possibility that affects it, we don't know. They used this Soraya assay, of course, on a heterogeneous population of cells. You amplify this, and you could amplify in this heterogeneous population of cells very rare alleles and see them. So I'm not sure, but whereas we use clonal, we use a mouse, right, which is clonal from one, and we didn't find with the same assay, we didn't find that what there's. So, and then third difference is that they use DNA, as what you do in cells. We used RNA, so DNA might hang on around longer, and you might get longer expression. Maybe there's increases cleavage. I think these are issues which are not resolved at this point.
So what does MECP2 do? So it's been studied for a while, connected to many repressor complexes, splicing, and now we find out it's an activator. So could you speculate on that? And does it sit on the rDNA clusters?
Well, I think it's pretty global. I mean, there seems to be not much specificity, right, when you look at this. And clearly it works as a repressor of some set of genes. I mean, there's no doubt about it. But more importantly, it works as the activator, or allowing gene expression, or maybe a high level of gene expression, probably by binding to the hydroxymethyl. I mean, this is really what, together with the Heinz data, I think they looked, that fits quite well, these data. So what it does, I think it's a very general, so I can't answer this.
Is there an emic connection?
Sorry?
Is there an emic connection? or direct MIC connection.
A MIC connection.
Because MIC is, again, quite general regulator.
So I'm not aware of a MIC connection with MSP2, and I don't know.
I have a quick follow-up question. I think MBD3 also binds to hydroxymethylation. Could it be that many of these methyl binding proteins have dual functions in repressing and activating?
Would be complicate our life, right? Yeah, so I don't know. So I think we have to see which genes, what's special about the genes which are inactivated versus those which are not, but rather probably boosted in expression. I mean, it's really not, it's not an activator in a sense, it really is a facilitator of expression maybe, right? It's more that than what MSP2 does.
The CRISPR system seems fantastic. So will or should the gene trap consortia change course and finish off the genome with the CRISPR system?
I would think so.
All right. If there are no more questions, let's thank Rudolf again for an excellent talk.